Hello, this is Dr. Robert Faust, and I'm here in my laboratory outside McMinnville, Oregon, in the wine country. And I'm doing an experiment here that I started last spring. I call this the humic acid torture test, where I take the worst soil I can find. I didn't have to look very far. One of my neighbors down in my farm in Independence, where we make the BioAg products, it's got some of the worst I've ever seen. It's a situation of abusing the soil. It's just constantly planting row crops. In this case, it's planting orchard crops, which, unbeknownst to them, soil was highly contaminated with pythium. They went and cleared it and planted new trees. Well, they didn't last that long. What a mess. But, you know, they never tested for anything. And those trees ended up getting bulldozed, and then they started planting corn. And in spite of the fact that I tested the field and found a dangerous level using the Becrop test, almost off the chart with pythium. It's just, they've been farming pythium. Yeah, they lost a lot of money on those other crops, and then they grow some like corn on corn. I mean, this is, in other words, this is farmland. It was some of the best in the world in the Willamette Valley. And it was deposited by flooding and had forests on it for a long time. And not long ago, you know, like 50, 60 years ago, maybe a bit longer, this ground was cleared from the forest and planted with orchard crops like cherries. And then over the years, disease took its toll. This, matter of fact, was the main area for growing hops. Pythium took its toll on hops. And then that pretty much went away, still out there. But anyway, so this is ground that's had nothing but chemical fertilizer, ever. There's no livestock operation. It's just spray and pray with pesticides and herbicides, a lot of them, which didn't end up working because they didn't really know what they were trying to control. But so it's just chemical fertilizer, lots of nitrogen, huge amounts, 200, 300, 400 units. The groundwater there is right up to the EPA max on nitrate. It's about 10 parts per million. So they've got, you know, really contaminated groundwater. And the crop yields are going down. What amazes me is the results that I've had in my own experiments on the same ground. But it's not so much the results and the science. It's a conditioning that they have. I mean, you can easily prove the point, which I'm going to do today with this little test. And anybody can do these little tests. So what I do is I take the soil, I screen it. Now here's an example. I screen it and I put it in a pot. This one's out of the pot. It's turned to a brick. This is the control. It just had rainwater, nothing else. And then I water with a specific concentration of humic acid. So I'm using full humics. In this experiment, I'm using full humics dry, actually, but this is the liquid form. So with the dry form of full humics, I'd be using 1 gram per gallon. That's it. So that's a nice coffee color in the liquid. That seems to be optimum rate for soil application. Any more is not any better, but frequent applications are better, but a big application is not better. It is homeopathic. It's a little is a medicine, too much is a poison. So you have to get the numbers right. So with the full humic, which is a guaranteed registered product, tested by the IHSS method and regulated by the State Board of Agriculture. Anyway, so you know what you got. And before, I mean, people know what they're doing. They really don't know how much humic acid was in something or how much to use, you know, and that's so crucial. Okay, so it would be like optimum if you had really high grade humics at 60–70%. It might really be 1.5 grams per 10 liters. Okay, so it turns out that a gram per gallon with this grade of material is going to be the upper limit. So gram per gallon in rainwater, which I collect for this. This is done completely with rainwater. So it's not biased to anything in the groundwater, which this groundwater here has got stuff in it. The rainwater is pure. Okay, so what we do then is we saturate till we get runoff at the bottom of the container, so that the soil is saturated. Okay, then the next step is we take millet seed and plant by hand millet seed, same amount per each container. I like to use millet because it's it's really resistant, drought resistant. A lot of people in northern hemisphere, Asia, everywhere use millet as a crop. It's a great feed for livestock, chickens particularly, but it's a human food, and it's amazingly productive. It's a grass, and it grow under horrible conditions, which is what I subjected it to here, really horrible conditions in a greenhouse, and then watering it only infrequently. I'm trying to duplicate the conditions that people would be growing it in, pretty much rain-fed situation, maybe with a little bit of irrigation, maybe not. So millet's a great indicator of humic acid, humic substance stimulation. Really, like the tests we did in China with wheat, millet, and corn showed the biggest yield increase on millet. It's very responsive, so I use it. It's the same millet that's in the bird seed you buy down at the hardware store. Usually it's a mixture, but you take the millet seeds out, just plant them. So that's why I use millet. I use millet because it's very responsive to humic substances. It's also very drought resistant, and it's short season too. It's like used in cold climates like Montana or maybe Canada, even Russia, China, etc. So we got millet here. So each one of these we're saturated, but with different concentrations. Okay, so we have a control. Here's the control, and it just had rainwater. So you can see it just broke off. The root system hardly developed. This soil just turned to pretty much to cement. But millet will still grow. There's the millet. Didn't produce any seed. So this would be the grower's soil without any additions. And we have the tests on this soil too, which proves that it's some of the worst soil that you're going to find. We'll get back to that one. So I'm getting kind of dirt on everything here. So then we have one that has 2 grams and 10 liters of full humic. That's number 2. And here it is, number 2. So there's number 2. We're going to weigh these up eventually. Now it's got enough root system to hold on, and it's got some biomass, and it's got some heads. It produced some millet grain. This is what you got. So that's the millet grain. Didn't do too well. Okay. Yeah, but it's still produced. Right. And then you have we also did fulvic acid. We have a famous product called Ful-Power. Fulvic acid, guaranteed, tested by the right methods. We know what it is. We know what the concentrations are. We've been doing it. What these test results are about on my millet trial, what does it all mean? This is absolutely new technology test method, and it tells us a lot more than we ever even imagined it existed. But what the exercise here is, and the crucial breakthrough that this is that the goal of converting a bacterial dominant soil to a fungal dominant soil with all the benefits of a fungal dominant soil can be done in as few as 90 days and get measurable results that show increase in fungal dominance and a reversal in many of the negative health factors of the soil, nutrition and health of the soil. It didn't, in this trial, it had rapid effect on soil dominance by fungi reversal. Now, that's just the beginning. That's the beginning of the process. That's not like instant, turnaround. It's still about a 3-year process to convert a depleted, compacted soil with lots of problems that you didn't even know existed, such as the properties of fungal and bacterial biocontrol. Biocontrol properties of the soil, the soil's ability to produce hormones or cytokinins that affect plant growth in a positive way, and also nematocidal properties, fungicidal, heavy metal, and drought resistance properties. Yeah, so this can be detected by getting deep into the soil food web, down to the DNA, and determine what's going on, plus other new test methods, and then analyzing it using artificial intelligence. So this is literally a quantum leap from our standard soil testing for NPK. It's just an amazing. And then what I'm showing here, using humic substances, activated humic substances, my Ful-Power humic acid dry, which is the most economical product on the market, when used at rates up to 2 grams per gallon, and a solution, and then saturating that soil at the right concentration. And that'll be explained later in part 2. But what we've seen here is what many people are looking for. We know that a bacterial dominant soil, and this happens over years, and when you break out the forest and start planting crops, you get deterioration. See the illustration on the preceding page. And then it takes years and years to build back that potential. And that's primarily due to organic cycling and formation of humic substances. Okay, so the soil gets harder and harder, bacterial dominant, and compaction occurs because the fungi help loosen the soil with mycelium and producing glomalin type materials that tend to bind, into a crumb structure. Okay, so then when that crumb structure breaks down, you have poor
Drawing my hair.
You have poor drainage and circulation in the soil. So that's why it's so important. And I'll go into more of that in detail, but this is what this test is showing. It's showing not only it's the same soil after about 90 days growing millet to maturity, and then testing, and we tested before also. We have it before, but this is comparing untreated, identical soil to the treated with a humic acid product, full humic, which is 60-some% humic and fulvic acid powder, soluble, to be used dry or liquid, and it can be bought as a liquid or dry or granular. So what it did, and at the at a fairly low rate. I mean, there's a specific rate above which there's no benefit, and below which you may be wasting your money. So there's a certain sweet spot, and we know what that is, and that was the rate we used. So then this one, I had many other test plots too. I did just fulvic acid and different rates and that sort of thing. But this is the one that really came out the best, because we also treated the seed with BioAg VAM, which is 7 species of endomycorrhizal fungi or ectomycorrhizal fungi. And yeah, so you can see here there's a rating here for mycorrhizal fungi, ectomycorrhizal fungi, buscular ectomycorrhizal ratio. So yeah, you see that it's very, very high, extremely high. It's because we added it and it reproduced. The other side is like essentially absent. So yeah, the ratio is just like the ratio of fungi to bacteria. We turned that around, really turned it around in the ectomycorrhizal area. And yeah, so this is the goal that a lot of people have, trying to use compost tea, things like that. But this is how to get it done, like in one season, and then improve the tilth and the productivity greatly, the yield. I mean, we improved the yield the first year in all these trials, no matter what we're doing, for the last, in my case, probably over 40 years. So that's the whole idea, is it takes time to rebuild that soil after years, 100 years or something, 150 years in this case of this soil, to degrade it. And it's a pretty resilient, pretty good soil to begin with. So to turn it around, I say in about 3 years is my experience. And, then it certainly pays off in water yield and all these factors that you can't see going on, nematode damage, fungal root rot, all these things. It's not just throwing fertilizer, NPK on it, and boom, the more you put on, the more yield you get. No. There's so many other factors. And this test also shows disease potential. It checks out by analyzing the soil with DNA, it can pinpoint specific diseases that are present in that soil that would affect that specific crop. And that's all super important. If that's there and you have very low resistance or a natural fungicidal or bactericidal activity, then you have a bigger chance of a crop disease problem. And you certainly should be thinking about that. So there's more to it. And I was licensed as a pest control production advisor called PCA in California, and it was insects, diseases, and weeds. I was licensed in those three areas, and so those are the three factors for sure: is nutrition, disease, and weeds. And this kind of testing gives me a bigger picture, certainly, and I'm looking at things in a very holistic way in terms of crop production and yield and economics as well. What's practical, what's doable. But this is actually a breakthrough, and that's why I'm presenting it. And go to part 2 if I do a part 2 on YouTube. So that was a test that I started last spring. It's on the worst soil I could find in the same soil type, 100 yards away, that wasn't quite as abused, but the same soil type, which was the Woodburn or Maribund series in the Willamette Valley. Abused soil, never had anything but chemical fertilizer for probably 50 years, maybe longer. And it was infected, and the trees died, and they didn't know why. Well, I know why, because we did a test and we came up high Pythium. But anyway, the point I want to make here at the end of this test, it's not over yet. We had to send in the Becrop data for total DNA of the whole microbiome, and we'll see what the difference is. But in terms of yield and soil structure, I did my first test with humic acids in, I think, in 1973 or so. And, you know, I get the same results every time. And now we have like full humic, which is water soluble, so then we know exactly what we got in terms of humic substances, percentage, based on a credible and certified test method. So we know what we got, we know what the optimum is. And then you add that to the poor soil, and you have a control, and then you see what the results are. What's the biomass, and what's the grain yield? Then you go on and you do the crush test and observe like the rooting, you know, amount of fine root, and then the structure itself, how it falls apart. And so the same results all the time. You get a drastically better yield, and I'll give you the numbers here. But bottom line, the control, which was really abused soil, it was hard, it turned into brick and all that. It's been, you know, plowed to death when wet. It's been packed. It's been hit with nitrates. And fertilizer, never had humic, never had organic matter, never had manure, never had compost. Okay, so the results are nothing. Here, you know, I mean, in other words, the control didn't even have any grain production. It had a small amount of biomass, and that's it. And then the full humic and VAM had a significant, considering it's a torture test, had significant millet production of grain millet and biomass, and the structure just fell apart. And versus the control, which is like big hard lumps. Okay, this doesn't surprise me. I've done it so many times, so many different soils. It doesn't surprise me. But here's what surprises me. That after all those years, you were 50 years, it's only just beginning to be accepted, that is humic acids, humic substances, within the conventional agricultural community. And I would say, in general, I mean, I would say that even the organic farmers weren't really accepting humic acids, you know, that much. And they had like all kinds of propaganda against it, like, oh, it can't possibly do anything. The only thing it's got is organic matter, and there's not enough to do any good. They didn't know about, organic functional group chemistry and activating pathways and soil microbial activity. That wasn't even anything then. It is now. But the results are dramatic, consistent, and economically an incredible use of money for input, especially on declining soil conditions. So I guess that's what I'm trying to get at, how it blows my mind that in spite of these kind of benefits that we're seeing, it's just not being readily accepted. Or if it is accepted, they've kind of bought some of the hype from the other people in the business. And the way that hype works is they'll show research and they show great results, but then they'll recommend a watered down liquid version, and they will recommend an amount that seems feasible to the grower. In other words, it's within their budget. And okay, so like a quart per acre or something, when you need 5 gallons per acre. So they're playing that game, and then the farmers don't see these kind of results. And then they say, well, humic acids don't work. Use the right amount at the right time, the right concentration, and know that it really is humic acid, that it really has the aromacicity and the organic functional group, polyphenols, quinones, and all that. And that's so easy to do with this a simple test like I did 50 years ago with sweet corn. I didn't know any of that stuff. All I knew, just like I know now, is that it grew better. It was bigger, had a better root system, and something happened to the soil structure. Did we know why? No, but it worked. It was an observation. That's the basis of the scientific method. And then working on it for 50 years, science developed, and then we started to learn. We started to learn how it was really working. So here we are, 2024, and according to studies, we may have 30 to 40 more crop seasons left before complete degradation of the soil. It's blowing away, it's washing away, they're turning it into cement. Compaction is the biggest limiting factor, and it's what we're doing, because it used to be horses and mules, and then a little John Deere or little farm-all tractors, little light tractors. Now it's gigantic things as big as a Bradley tank, or they're huge, heavy equipment pulling 50 foot of heavy harrow and compacting like trucks running semis out there on the harvest. So it's just compaction and tillage and compaction, beating it fine and then compacting it, and then it gets wet and turns to cement, and you go to town and get a job. And that's what's going to happen. That's what has been happening. And yet, for a few dollars per acre, even if it was $100 an acre, it'd certainly be well worth it. We generally see a 6-to-1 return, like $6 back for every dollar spent with humic acids. But I mean, I don't guarantee that. Everybody's going to have a different result depending on their practices and where they are and everything else. But basically, we've demonstrated, and we're going to demonstrate more using high-tech Becrop testing, just what happens in terms of the microbial basis. I've already shown in a scientific paper that we increased fungal biomass in 60 days with using humic acids over control. Now we're going to get more specific, because that was a primitive test by that just shows basic biomass of bacterial and fungal. Now we have something that's really advanced and really get down to a database with 24 million different species. So it's really going to show something. But in spite of what I can show, either I don't know whether people don't believe me. There's something to that. They've been brainwashed mainly. I mean, they're listening to the farm, so-called farm advisors that work for the chemical companies. So it's really the message. It's really a diluted message that confuses the growers. And they don't know what to believe. Bottom line, they don't know what to believe. They should believe me. I got nothing. I got no reason to say anything but the truth. I'm 78 years old. I've been working on this since I was in my 20s. The results are clear. The benefits are obvious. You can do these little tests yourself. That's why I did the test using millet, because you just pick it out of the birdseed that you buy down at the hardware store. Millet's a great, quick, and a great indicator of fulvic and humic acid activity. It's very, very reactive, very responsive. So that's why we use it. So is sweet corn. It's just millet's, I don't know, it's just, it can take the torture test better. And that's what I'm trying to do here. Like if you were in a subsistence farming situation up in the north, dry land, and you're growing millet, and this is what you'd be facing. And so with a small amount of humic acid, we demonstrated that in China with 4 ounces of humic acid in the row. And we saw a better than 50% yield increase. That's the Chinese Academy of Sciences, Agricultural Sciences. But anybody can repeat this test with your own soil and find out if it's true. You don't have to believe me. Believe your lying eyes, you know. There it is. That's all you got to do. It's a controlled and treated, and come on. This can change the world. I mean, not only does it increase the food production, increases the soil health and biomass, and it's also helping reduce CO2 levels, because as you increase the sugars because of carbohydrate production by photosynthesis, that's using CO2 from the air to convert to sugars. Excess sugars are excreted by the roots, and the microbes, especially fungi, feed on that, release phosphates that help the plant grow better, to produce more sugars to help the soil rises for your organisms. You don't have to add sugar. The plants produce their own sugars. You just have to give them what they need, and they need humic substances to catalyze, to increase chlorophyll production, the little structures that convert CO2 to sugar. And they produce the sugars. It's ridiculous to take sugar from another plant, like a sugar beet or sugar cane or whatever, and add it to another crop to benefit them, when the problem is a lack of the ability within the plant, within the soil biome, to complete that task and reduce the CO2, taking it from the atmosphere instead of releasing it. If you add sugar to the soil, what happens first is yeast, which is everywhere. And yeast converts sugar or sugars to alcohol or vinegar, well, alcohol then vinegar, okay, and releases CO2. So instead of sequestering CO2, it's releasing CO2 when the yeasts convert it to alcohol and CO2. I mean, if you've ever fermented something or made beer or something, you get CO2 bubbling out into the air. And you get alcohol, which if you put enough sugar and you get enough alcohol, it sterilizes the soil. So this is the advice that people are getting. Let's use sugar. No. Let's use humic substances that help produce sugars, help the plant produce sugars by taking CO2 out of the atmosphere and converting into sugars, which is metabolized into microbial proteins and humic substances. You're actually priming the pump. You're using humic substances to catalyze, to stimulate additional production of humic acids. So I call that priming the pump. And that's the answer. And, you know, all the tests that we do, you know, well, you know, you'll see the rest of the video, that, or the previous part that shows the results. And so this is the way we get to really tune it in. Why is the fungal to bacterial ratio of soil so important as an indicator of soil health and tilth? As you can see from the chart on the left, it shows the progression or succession of plant growth, and it culminates with high canopy forests going through 5 distinct phases. And really, agriculture in Europe, Northern Europe, and most of the world is dependent on going into areas that are forested, clearing them, and farming. And then also areas of prairie, which have tens of thousands of years of dying and decomposing grasses and manure. But that basically, you go through the same succession though. And in many places that I've been where they use slash and burn methods, it's like 1 year of corn and beans, and then up to 20 years of rainforest before you can get optimum yield. So that's the thing. When you break down this cycle of the deep-rooted plants bringing up minerals that have been leached deeply in the soil and then releasing a lot of biomass that decomposes, and especially the hardwood trees, to produce humic substances and feed the soil micro needs. The fungus need organic matter to feed on. So once organic matter is low, that's going to be lights out for all these beneficial fungi that have such an important job, really. I mean, the fungi helps protect plants. There's fungal diseases, but they're weak, unspecialized, and so you get fungal production of fungicidal materials, and that's crucial to the balance, and even controlling nematodes, etc. So that's why things go downhill after they clear forest and start growing corn, and then of course you move west, you keep moving. Now there's no other place to move, so we have to understand the principle here. I mean, it's soil mining and moving on, which has happened to entire civilizations over and over again. But it's either understand the mechanisms and maintain and build the soil, or go the way of many other civilizations that came and went, and you can find their ruins of their civilization out in the dry area of the Pacific Southwest, and it goes right back to destroying soil structure, and then the wind and the desiccation, and it just breaks the cycle, and you end up in desertification. So this is really it in the most simple form. It's the hardwood tree biomass feeding the soil fungi. Now, we did that in 90 to 100 days by adding concentrated humic substances, which was actually from an ancient forest that got buried probably in the last flood in the Ice Age, and then composted for either millions or tens of thousands of years, depending on the deposit. So it's nothing more than fossil compost that's highly concentrated. It takes 9 to 12 tons of compost to equal a pound of the soluble humic acid powder. So it's an economic choice. And of course, compost is another choice, but you're talking about large tonnages. And so this test proved, what's going on. We proved that with applications, annual applications, to get a kickstart, you can restore a soil quite rapidly, at least in soils that were pretty good to begin with, like the soil that I was testing here in the Willamette Valley. Let's say a degraded soil. And, but, so it's really the answer of answers, because without a steady food supply and what the soil can supply, all the rest isn't probably going to matter. That's the irony. The simplest thing out there that people don't think about is the soil and soil productivity, that the entire civilizations depend on it, and that we have, based on studies, credible sources like MIT, we have 30 to 45 years or crops left before it's either a complete wipeout or financially, economically not viable. So we know the end point if we don't do something about it. And bottom line here, I'm demonstrating the method to deal with it. And there's several methods. This is one method. And we've shown that it will turn things around dramatically in terms of fungal dominance. It's really just an amazing thing to see happen. And this is the first time we could really measure this and all the other effects that go along with it. So stay tuned for part 3, and I can get more into detail, but I think this chart here explains a lot of it.